Chapter 13, Topic 3.2, Stage 1, the development of lymphocyte diversity. In this topic, the learning objectives are: summarize the maturation process of both B cells and T cells, explain how our bodies are equipped with lymphocytes capable of responding to nearly any antigen imaginable, describe the B cell receptor and the T cell receptor, outline the processes of clonal selection and expansion. So in this topic, we're going to talk about specific events in T cell maturation, specific events in B cell maturation, the origin of immunological diversity, and clonal selection and expansion. T cells mature in the thymus, a small gland situated behind the sternum or the breastbone. Once the T cell leaves the thymus, it has some special molecules that are involved in antigen recognition or communication with other immune cells. One is cell determinant, or CD3. It supports the T cell receptor and other specific molecules. In T helper cells, they support CD4, like in this diagram. Cytotoxic cells have CD8. Remember, CD4 equals T helper, CD8 equals cytotoxic T cells. B cells, on the other hand, mature in the bone marrow. When they are finished maturing, they will have a B cell receptor on its surface that is basically the antibody they are capable of producing. It's like a big old saying, "This is what's in my store." B cells also have MHC2, but more about the B cell receptor / antibody of the B cell. Each B cell can produce antibodies against every shape of protein or other macromolecule that can come along. This diversity of antibodies arises from shuffling of different genes that code for the variable region of the antibody. These genes get shuffled during development so that, hopefully, you get all of your B cells coding for different antibodies. This gives you a wider potential immune response. T cells undergo a similar shuffling of variable region genes, but their T cell receptors are never excreted. As T cells and B cells mature, the body eliminates all cells that have receptors for self-antigens. You don't want your immune system attacking your cell, now do we? B cells and T cells that don't have self-antigen are allowed to mature. These mature, naive cells wander around looking for their antigen. So, here's a triangle-shaped antigen. It fits into the variable region of this particular cell. The cell goes, "Woohoo! It's time for me to go spring into action." It begins to divide so that there's a whole lot of it to go into battle against the pathogen that carries that antigen. The other cells watch with envy. But we don't need a whole bunch of cells if we never encounter that particular pathogen. If I never go to Africa, I will likely never encounter malaria. So, why waste energy making a bunch of malaria clones? If I lived in Africa, the same would go for pathogens unique to our area. Well, that's it for this topic. In the next topic, we will cover the presentation of antigens to B cells and T cells. In the meantime, here are the learning objectives for this topic. Summarize the maturation process of both B cells and T cells. Explain how our bodies are equipped with lymphocytes capable of responding to nearly any antigen imaginable. Describe the B cell receptor and the T cell receptor, and outline the processes of clonal selection and expansion.